Okay, so today we're going to focus on what happens at the end of the neuron and how do we get that signal to go from one neuron to the next across that synaptic gap. All right, and then we'll talk about the myelin sheath a little bit. All right, so this is just a little bit of a review on your action potential. Out here I have the positively charged outside of the neuron and the negatively charged inside of the neuron. Generally, we go through and we focus only on one membrane, but remember that we have both sides of the neuron that are doing the same thing, so it's positive out here as well. All right, the pink boxes or lines right here are representing the sodium channels, and the blue are representing the potassium ion channels. Okay. If we remember, we have lots of potassium intracellularly and lots of sodium extracellularly. Okay, so as we have progressed through this axon, my action potential has moved on down the axon. By the time I get to this area, we know that the sodium gate is going to open because the change in charge is going to be taking place from farther up. So as this action potential, the intracellular area, kind of starts to come down to this section and hit these channels, then the sodium gates are going to open. Sodium is going to move into the cell. Once it reaches a certain length of time, okay, it's open for about 1 millisecond, then it's long enough to change these charges so that the outside becomes negative and the inside becomes positive, which will trigger the potassium channels to open. Potassium then will move out, which is going to revert it back quickly, which will then trigger the next set of sodium channels to open. They will move down, switch the charge back again. Potassium is going to be moving out, which is going to switch the charge back again. So we continue on in this fashion until we get to the end. All right, in the meantime, as these charges are taking place and moving down the axon, keep in mind on this diagram I do not have the pumps, but the sodium-potassium pumps are working to reestablish that resting gradient or that resting potential. Okay, so what I want to do today is I want to focus in on the end or the axon terminal of the cell. So we have another little ion that's going to come play a role in this today, and that is going to be the calcium ions. And remember, good sources of calcium are any of your dairy products, so it's super important that we make sure that we're drinking our milk or eating our cheese and yogurt, because the calcium really not only is going to help to build strong bones, but it also helps to make sure that your nervous system is functioning properly. And here's why. Calcium-gated channels are also voltage-gated ion channels, as we've seen so far, where it is a change in the membrane potential that will open and close the gates of the calcium ions. Calcium is found at the end of the axon, okay, so at the axon terminals, and that is the only place that we see these particular channels. All right, so as our action potentials move down the axon, when this voltage change comes down and we reverse the voltage here, that is going to stimulate the calcium ions channels to open. When the calcium channels open, calcium then is going to rush in to the intracellular area. So they will be here into this terminal. All right, as calcium moves in, what we see happen is that calcium is actually going to come over and bind to these vesicles. And so what I have in the axon terminal are I have different neurotransmitters. These neurotransmitters are made of proteins. Remember those amino acids that we saw floating all the way through here on the intracellular section? Well, through protein synthesis, they have been incorporated into a three-dimensional protein structure, and they are wrapped in a plasma membrane, okay, right here. And inside that plasma membrane is our neurotransmitters or nervous system hormones. Okay, so we have like acetylcholine, serotonin, not epinephrine, nor what's the other one that I'm thinking of? dopamine, serotonin, I think I said that one. But these are some of the neurotransmitters that are found here in your nervous system. All right, so they're wrapped in these nice little sacs. And so once the calcium ions bind to these neurotransmitters, it is going to cause the neurotransmitters to move over to the end, and they are then going to bind with the plasma membrane itself. Okay. Now, we did a little demo of this when we stood in a circle and I had the volleyball in my hand. And because the surface of the vesicle is made of the same material as the plasma membrane of the neuron, it makes it very convenient that through the process of exocytosis, that the membrane can fuse with the neuron, okay? And once it fuses with the neuron, let's see if I can go, it opens up, and these neurotransmitters then are free to come out into the extracellular environment, okay? All right, so now I have these neurons in this junction area, or in the synaptic gap. So what happens next? Well, once they are in that synaptic gap, their job then, I'm going to move to my next neuron. I need to go up. Make sure I got the right direction. There we go. All right. So here's my presynaptic neuron. Okay. And I've got my neurotransmitters that have been released into this gap. All right, well, the neurotransmitters are going to come over and they are going to attach to these receptor proteins. Okay, these red dots are fitting very nicely into my receptors. Okay, if you can tell by the drawing, these receptor proteins are attached to my pink channels. Now remember, my pink channels were the sodium channels from before, and they are gated, which means that they are not open until they are stimulated to be open. All right, so now I am at my my postsynaptic neuron, so I am after the junction. Okay, and so what we want to do is we want to open up these sodium channels so that we can cause this action potential to move through the dendrites, down the cell body, to the axon terminal at the end of this one, and then continue on till we get a response. All right, so what do? Well, because this particular sodium channel is not being opened by electrical current, it is being opened due to the binding of a neurotransmitter. We now call this a ligand-gated channel protein. All right, so this is a ligand-gated channel protein. Remember, ligand refers to any molecule that is going to bind to a receptor to cause a response. All right, so we have a neurotransmitter that is binding to a receptor. It is attached to a channel protein, and so when our neurotransmitter binds to this receptor, it now will open the gate and allow sodium to move in. All right, so now we're going to start this process over again because sodium is rushing in. We're going to see the membrane potentials change. Because the membrane potentials are changing in this area, that change in potential is going to cause the potassium gates to open. All right, so crazy stuff. What is going on here? So we have what's starting our process here then is that we have a ligand-gated channel protein. Then what we do is that because we're allowing sodium to switch places, we now are going to switch back when we get to potassium here to a voltage. Gated protein channel. Okay. Now, once the potassium rushes out, okay, this is the repolarization of my action potential. Our pumps will go back to reset everything as normal, and this next group of sodium channels are going to open due to that voltage gated. So what we've seen already is that we've seen the voltage gated channel ions are active most of the time throughout the neuron. We see the ligand gated channels are really in the dendrites of the nerve cells, which are going to really get this kind of party started. All right, and so we kind of continue on and on and on. Now, these neurotransmitters here, keep in mind that all they are doing is attaching to the receptor proteins. They are not entering into the cell. They are not doing anything else. They really are just binding to this receptor. If we don't have enough spaces for these neurotransmitters, a couple of things can happen. We have enzymes specifically designed to destroy neurotransmitters. Okay, so some of them are destroyed. Okay, they're broken apart. Their parts, which are their amino acids, are going to be reabsorbed so that they can be incorporated into other neurotransmitters. Also, though, what we see is that some of these neurotransmitters will actually come back and bind to the presynaptic neuron. And this presynaptic neuron will actually fold in on itself. I'm not sure how I'm going to animate my drawing here, but it will fold in on itself and it will hold them back in so that we can then reuse those neurotransmitters again. Okay, so some of them are degraded out here in this synaptic gap area, and some of them are reabsorbed through endocytosis. So that we can reuse them. Okay, and that is how we go from one neuron to the next neuron, and then we can go back and continue on until we get to the end of our nerve cells and cause a reaction. Okay, so now I'm going to switch back to the slides here, so I gotta move my screen a little bit, sorry. Where'd I go? Nope. Okay, I'll figure this out in a minute. All right, so I have switched to my screens here, and so now I have my slideshow of basically the information that I just drew out for you. So let's go through this really quickly here, just kind of as a little bit more formal piece of notes. All right, so we have a chemical synapse here. So at the end of our axon, the axon terminal right here, here are our vesicles that contain those neurotransmitters. Okay. At the end of the synapse, what we see is that the action potential will depolarize the membrane, causing the calcium channels to open. The calcium channels are going to bind to these neurotransmitters vesicles right here. Okay. The neurotransmitters will fuse. They come down and they're going to fuse with this membrane and release these neurotransmitters into the synaptic gap area. These neurotransmitters then are going to diffuse across that space, and they are going to bind to the protein receptors on the postsynaptic neuron. We can see the little red dots right here. Once these neurotransmitters bind to these proteins, they're going to open up the gates, allowing your sodium ions to rush into the cell, changing the membrane potential. Then we'll see the potassium ions rush out, and then we'll see that whole chain reaction, which means that we are now switching back to that voltage-gated reaction as it moves down the rest through the dendrites and down the rest of the axon. All right, so these are your gated channel ions will open in response to that. So remember, we've switched back from a chemical, now back to the electrical gradients. Neurotransmitters will either be destroyed in this cleft area, or they will be reabsorbed into these vesicles and then reused. Okay, you can see this chemical signal coming in, and again, you can just see how it's a nice animation of how this actually works. Signal comes down, calcium gates open, bind, they release neurotransmitters into the area. You can see these neurotransmitters binding here to our ligand gated channels, causing the exchange of ions to now start to take place.